Hello, hi everyone. Today I have come to talk about the last chapter that is included in class 10th CBSE portions that from NCERT book, that is Heredity and Evolution. In the chapter Heredity and Evolution, only the heredity part is included from the recent syllabus. The evolution part has been removed. So we'll be talking about heredity, and in the last I'll be talking about a little bit about sex determination. So let's start with the chapter Heredity and Evolution. So to start with, when we tell or we talk about the term heredity, it's a very common term which we use very frequently. In any day we want to refer to some traits or features about from our grandparents to the grandchildren or the parents to that of the children, we tell it's a heredity that has been passed on. We mostly tell it is the heredity. So when we want to define heredity, exactly what does heredity mean? Heredity is defined as a particular characteristics or trait or feature which is found or which is observed in an individual or in an organism, is passed from generation to generation or is passed from the parents to that of the offspring. So let's see what is the definition of heredity. It is defined as the characteristic by which the traits, features or characters of a living organism is passed on to its offspring from the previous generation or passed on to its offspring to that of the next generation. Now in heredity, when we are talking of heredity, we are using a term trait. We are using a term trait. Exactly what does trait means? What is it that traits make us understand? In scientific terms, when we talk of traits, it is an observable feature or observable characteristic which or a particular condition is called a trait, which is found in any individual or any organism. So an observable feature, characteristic or condition is called a trait. Anything which is visible or a particular feature which is visible is known as trait. Now traits are again of two types, that is inherited traits and acquired traits. Inherited trait means those which have been inherited or which have been given or which have been transferred from the parents to that of the offspring, which have been got from the parents, some inherited traits. Like we tell that particular boy a similar kind of hairstyle, that we tell has similar hairstyle to that of his father. That is an inherited trait that has been passed on from the father to that of the child. Sometimes the skin color, skin tone, eye color, that all are kind of inherited traits which is passed from the parents to the offspring or from the grandparent to that of the offsprings. And the other is the acquired trait. What is acquired trait? The name suggests it has been acquired. It is not that we get it from our parents. It has been developed or it gets developed for a particular person, and it develops due to the external environmental factors. That is writing or supposedly kind of swimming. Those are acquired. It is not that it is transferred from the father to that of the son or father to that of the daughter. It is been acquired by that person for its living or for the external environmental conditions. So let's get to know a little bit in detail about inherited and acquired traits. The acquired traits, they are the characters or features that are developed by an individual in response to the environment and its surroundings. So when a person or an individual is staying or living in a particular surrounding, due to that particular surrounding or environmental factors, they develop some traits, and those traits are known as acquired traits. The genetic change or DNA alteration doesn't take place. There is no change in the genetic constitution due to a particular acquired trait, or acquired trait does not harm the genetic constitution of a person or an individual. It remains intact. It is just that the person or the individual gets to know or starts to do or gets to understand some skills or some knowledge for the surrounding in which he or she is in. The characteristics are not transferred from one generation to another, which I already told, and the traits are involved like that of cycling, driving, swimming, reading, etc. So these are the traits which are acquired by the person. So these are not passed from one generation to another. And one more important thing, it does not bring about any genetic constitutional changes in the person. Now let us get to know about inherited traits. Inherited traits, they are the characteristics or features that is taken up from the parents by their young ones. It is brought about due to a change in the genetic constituent or DNA of the germ cell. So particularly this characters or inherited traits is present in the DNA. Because of the particular gene or the constituent or the constitution of the DNA, this trait is seen or is observed. It may be a skin color, it may be the hair color, it may be the hair type. It is transferred from one generation to the other, like that of the skin color, hair texture, height, eye color, etc. They are all examples of inherited traits. Now, what are traits, or why are traits inherited? When we talked that, we were telling that okay, traits got inherited or traits were being taken up from one generation and passed on to the other generation. But why does that happen, or how does it happen? Why does it happen? So when we talk about it, is the rules or the laws of inheritance, because our genes, the genes that we are talking about, which carries these inherited traits, follows the law of inheritance. And these laws of inheritance was first given by or laid down by the father of genetics, that is Gregor Johann Mendel. So Gregor Johann Mendel, he was the person who was also termed or is also known as the father of the genetics. He was the first person to formulate the laws of inheritance. Before formulating these laws, it was he who had studied this, how do these characters inherit from one generation to another. And he had done all these experiments in the sweet pea plant or garden pea plant. It is a Pisum, it is scientifically known as Pisum sativum. And he had done these experiments and he had studied in 7 contrasting characters for this pea plant. Now, why did he select a pea plant, or why did he select Pisum sativum? He could have selected anything and anything that was there. He could have observed it in humans, he could have observed it in animals or any other things. But why did he select plant, or that to a Pisum sativum? Now let us get to know the reasons behind why he selected this pea plant. They have quite a number of observable traits or contrasting characters, because he wanted to study how the characters or the traits are inherited or how they are passed on from one to another. So he had to study many traits. It was not that 1 or 2 traits he was keen on studying, so he had to study many traits. And this garden pea or Pisum sativum had many different characters, different observable characters. Observable characters means which can be very much observed from the naked eye. That means it can be the flower color or the stem length, or it can be the pod shape, the seed shape, the seed color, which is very easy to study and which is very easy to generate also. The things to visualize is very easy. It is very easy to take the data or take the readings of it. So he thought of this pea plant because this was having many contrasting characters. The plant was easily available. The material also has to be easily available, where he can get the pea plant from anywhere. So it was available material. It was convenient for Mendel to control the mating of the plant. Because every time he wanted to study the inheritance pattern, every generation he had to undergo hybridization. He had to cross those plants to develop a different offspring. So it was easy for him to develop the crossing or to undergo the hybridization experiment. So it was easy for him, the plant short lifespan, so he could study quite a number of generations. In order to study the inheritance pattern, or in order to study the inheritance, he had to see multiple generations of the particular plant sativum. It actually completed its life cycle in a very short span of time. So he could study its changes or its pattern in multiple generations. It is not that 1 or 2 generations. He can study multiple generations in his lifespan. So it was easy for him to do. And one successful cross, and after crossing, the cross seeds that he got, it was quite in number. So all these characters or all these features were very important, which he had to think of before starting of the experiment or before starting of its inheritance, or to study the pattern of inheritance. So these were the 7 contrasting characters based on which Mendel did his experiment. These were the 7 traits which he studied, and these were very detectable, observable, visible traits for the human eye. So this was very easy for him to detect the changes and to take a note of those changes, based on which he later formulated the laws of inheritance. So all these what were seen, all these characters are dominant types, and all these characters were of recessive type. So these characters, these 7, that is tall, eggshell, purple, green, inflated, yellow and round, are dominant type of the characters, and these are the recessive type. The dominant type means it always dominates, it expresses itself every time wherever it can. So when we talk dominant trait, it is the trait or the characters that expresses itself in the absence or presence of the recessive trait. So dominant trait or dominant trait is something that will express itself. If it is there, immediately it will express itself. It does not bother whether recessive is there or not there. It is there or it is absent. It doesn't bother. Dominant is there means immediately it will express itself. Whereas recessive, on the other hand, is a subdued one. It expresses itself only when the dominant is not there, or it is not able to express itself in the presence of the dominant trait. So recessive trait is a trait or the character that expresses itself under homozygous condition or in the absence of the dominance gene. In the homozygous condition, this is another term that we are coming out to, that is homozygous condition, homozygosity. What is homozygous? So, and under the absence of the dominance gene. So we should remember that dominance expresses itself, or it is a particular trait or the character that expresses itself in the absence or in the presence of the recessive trait, whereas recessive trait is the character or that expresses itself under homozygous condition or in the absence of the dominance trait or the dominance gene. So now let's get to understand what is homozygous and another term, that is heterozygous. What is homozygous? What is heterozygous? Now when we told under homozygous condition, homo means same. Homo means same. Hetero means different. When we tell homozygous, that is the zygote, that is the zygote that is there, constitute the same genes. If you remember from sexual or how do organisms reproduce, we talked of the zygote. The zygote contains the male and the female. The union or the fusion of the male gamete and the female gamete gives rise to the zygote. Now when we talk of homozygote, homozygous, that means the zygote contains, so we can understand the zygote means it will contain a pair of allele. It will contain a pair of allele. Now that pair of allele in the homozygote condition is same. That allele, both the alleles, I told you a pair of allele, that pair of allele will be same. That is, it will be capital T means it is same. That pair will be same, exactly same. Capital W or small t. This is a dominant trait and this is a recessive trait. In whichever condition, it is the same alleles that is present in the zygotic condition. So it is called a homozygote or homozygous. It is homozygous. That is, it is having capital T, small t, or small w, or capital W. So what we can describe or what we can tell about homozygous? Homozygous is a genetic condition where an individual inherits the same alleles of a gene from both the parents, or when we refer to a homozygous condition or a particular trait, it implies both the alleles for that trait are either dominant or recessive. That trait contains is either in the dominant form or in the recessive form. But both the alleles are same. But when we talk of heterozygous, hetero means different, and zygous means in the zygotic condition. So in the zygotic condition, if it is different, that is one is dominant and one is recessive, then it comes as heterozygous condition. That is capital T small t or capital W small w. I hope it is clear. It is in the same allele, whichever genes are there, they are in the same position, or they are in the same condition. That is, if it is dominant, both are dominant. If it is recessive, both are recessive. Heterozygous means the allelic pair is different. That is, one is dominant and the other is recessive. One is dominant and the other has to be recessive. So heterozygous is a genetic condition where the individual receives two different alleles from the two parents. Here they receive the same alleles from the parents. Here they receive the two alleles. In a heterozygous condition, one of the alleles constituting the trait is dominant and the other is recessive. So one of the trait is dominant and the other is recessive. So obviously in a heterozygous condition, the dominant trait gets expressed, because I told that recessive cannot express in the presence of dominance, whereas dominance expresses every time whenever it is present. So whenever it is there, it will express itself. So in the presence of dominant and recessive, always the dominant gets expressed. Talking about homozygous and heterozygous, I told you the same pair of allele or different pair of allele. I told you this is the same allelic pair that comes. So what is an allele? So what is an allele? An allele is a different form of a gene, is known as allele. Imagine a gene may be for your height of the plant. So we can mention it as T. It is because it is the height of the plant. It is tall and dwarf. It can be of the two types. So we can tell that capital T is for tall plant, small t is for dwarf plant. Now, different forms of the gene. Now, alleles are those T that what I am telling, that is allele. It is the different forms. Because it is different form means one is capital T and one is small t. Both regulate the height, but they are in different forms. When it is capital T, it is in dominant form. When it is small t, it is in recessive form. So, allele is the different form of a gene, is known as allele. And next we come to genotype. Or we'll be talking, or let's understand the term genotype, because when we will talk about the about Mendel's cross inheritance laws and all, we'll be talking about genotype and the phenotype. So what is genotype? So genotype is a genetic constitution. It deals with the genetic constitution of an organism or of an individual. It is the genetic constitution. We have to see what genetic constitution is there. Whereas phenotype is described as the visible or detectable traits or characters in an individual or an organism. It does not refer to the genetic constitution. So genotype is a genetic constitution or an individual. In general, genotype is referred to the genetic make of the particular character or the trait. The genes present determines the visible characters of the traits. Whereas phenotype is described as a visible and a detectable traits or characters in an individual, and it arises due to the interaction of the genotype as well as the environment. It arises due to the interaction between the genotype and the environment. So phenotype is the observable features, whatever we are able to see. We see a tall plant. When we see a tall plant, we say the plant is tall. But we don't get into it to understand whether it is capital T giving a tall plant, or it is capital T small t giving a tall plant. Because both gives a tall. Because it is the presence of the dominant genes, gives a tall. But whether the dominant gene is present in homozygous condition or it is present in heterozygous condition, we are not able to understand from the phenotype. Then we have to refer to the genotype of the character. So now let's understand what were the experiments, or how Mendel did his experiments, or what actually he got from his experiments. So the first thing that he did was he did a monohybrid cross. So the first thing so now let's understand the monohybrid cross. This cross was formulated, or this cross was first done by Mendel, and this cross was, as the name suggests, it includes only one character. Only one contrasting character is taken into consideration, and the cross was done in order to represent, in order to understand the offsprings, how the offsprings will be, or what will be in genetic makeup, what will be its phenotypic makeup. So this was taken, and in this plant nothing was considered, only the height of the plant was taken into consideration, and it was done. Now one is a tall plant and the other is a dwarf plant. Now these two plants were crossed. These two garden pea plants were crossed. So when they were crossed, so one is a tall plant, that is pure tall, that is dominant, capital T, and this is pure dwarf, that is small t. You can understand, or you can understand that when I'm telling of a dwarf plant, that plant has to be a pure dwarf. It has to be small t, because when I told you, if the dominant gene is present in the plant, immediately it will give a phenotype of a tall plant. So because it is dwarf, it means that the plant is small t, it is pure recessive, and this is pure dominant. Now this capital T and small t, capital T is coming from the dominant or the tall plant, and small t from the dwarf plant. These two are hybrid, these two combined to form a zygote, that is small capital T, small t. Now this capital T, small t that is formed, it is a tall plant. It gives a tall plant. This capital T is capital T and small t is referred to as the parental generation. We call it as the P1 generation or parental generation. Now when they are crossed and they give a tall plant, that is a capital T, small t plant, that is, it is a heterozygous plant. We call it a F1 generation plants. We call it a F1 generation plant, that is their hybrid plants. They are hybrid, they are heterozygous in condition. Heterozygous means as we have crossed and developed, so it is called a hybrid. Because we have hybridized it, because we have crossed it, so it is called a hybrid. And these plants are 100. All of these plants have the James genetic constitution, that is capital T, small t, and this is a 100% hybrid. Now this capital T, small t will produce gametes of one capital T and one small t. Again, the other plant will also be producing one capital T and one small t. Now when this capital T, small t and capital T, small t are crossed in all possible combinations, that is, see, capital T can come with this capital T and form capital T, capital T. This capital T can cross with this small t and come capital T, small t. This small t can cross with this capital T of the other plant and come capital T, small t. And small t of the first plant and for small t of the second plant can cross, and they can come to small t, because it is recessive plant. This will be a dwarf plant. So this will be a dwarf plant. See, in this is called a checkerboard, or this is called a Punnett square. This is called a checkerboard or the Punnett square. When like this, if we are doing for one particular trait, it is easy to understand or it is easy to do. But when we are doing of multiple traits, that is 2, 3, 4 traits, when we go, the number of plants starts increasing. The number of options, that is in all possible combinations you have to do. Here from 2 gametes we give rise to 4 plants. So immediately it starts increasing. So that time it is difficult to manage out this way, because always there is a chance of doing a mistake. So we always do, or we always refer it, or we always try to do it in a checkerboard, or this is called a Punnett square. So this was the capital T gamete that was formed from the pure tall plant. This was the small t gamete that was formed from the pure dwarf plant, and it gave rise to capital T, small t, which is a hybrid or the F1 generation. Now this capital T, small t, these plants, this is a capital T gamete we call, small t gamete, capital T gamete, small t gametes. These are the gametes, and this was done on the selfing, because basically they were selfed. Self means the pollens of the flowers were put on the flowers, on the same flowers. So they were not put on the other flower, they were not crossed. Crossing, when we are doing crossing, it means that the pollens from one flower or from flowers from one plant is taken and it is put onto the gynoecium or the carpel of another plant or flower from another plant. It is totally different plant. But here selfing means the pollen from the plant or from the flower falls onto the carpel of the same flower. So this goes to selfing. When we talk of selfing, so what happens? These are the gametes that is formed, capital T, small t, capital T, small t. Now if you put it in a checkerboard, so this is the sign, or this is the sign of a male, that is capital T, small t. This is a sign of female, that is capital T, small t. Now we will write all the combinations that we get from this checkerboard. So that is capital T, here capital T, capital T. That is capital T, small t comes, it is heterozygous tall. Heterozygous, but it is tall because the presence of the dominant gene. It is homozygous tall because both the genes are capital T. Again capital T, small t, that is heterozygous small, and this is homozygous dwarf, that is small t, small t. So when we look at all these four plants, we get, what do we get to see? We get to see this is a tall plant, we get to see this is a tall plant, we get to see this is also a tall plant. So we get to see 3 tall plants and 1 dwarf plant. So the phenotypic ratio, or the ratio or the plants which we get to see on based on what we see, we can tell that it is 3 small plants, 3 tall plants and 1 small plant. That is phenotypic ratio is 3:1. But when we look into the genotype, or when we look into the genetic constitution of each of the plant, we get to see that capital T, that is homozygous tall, is only 1 plant. Heterozygous tall, that is capital T, small t, there are 2 plants. And small t, there are only 1 plant. So 1 is a homozygous tall plant, 2 are heterozygous tall plant, and 1 is homozygous dwarf plant. So this we can constitute or tell it as the genotypic ratio of 1:2:1. This is a monohybrid cross. This is very simple as we deal with only one character for the cross, and one character is kept in mind and we talk or think about when we take the data. It is only of one character. Now Mendel performed another experiment. In this he kept in mind, or he wanted to think of 2 characters at a time. So what did he take? That was a round seeds and yellow seeds, and this was wrinkled seeds and green seeds. He crossed the plants with round and yellow seeds along with plants of green and wrinkled seeds. So what did he get? When he crossed with round and yellow seeds and green and wrinkled seeds. So round was given the genotypic constitution of capital A, whereas yellow was given the genotypic constitution of capital B. Whereas small a was for green in color and small b was for the wrinkled in one, or whatever. Capital A for the round and small a for wrinkle, capital B for yellow, and small b for green in color. So when they were crossed, or when they were hybridized, just like that in the monohybrid cross, they developed a plants having the genetic constitution that is heterozygous, yellow and round, that is capital A, capital B, small b. Here because the dominant character of round seed, the dominant character of yellow seed, both are present. So it is capital A, capital B, small b. Now these plants, again they were selfed. So when they were selfed, pollen from one and pollen of one, and the ovum or the ovule of the same, in the same flower it was done. The pollen was dropped into the ovule of the same flower, within the same flower. So what happened? So the genetic constitution of the pollen as well as the ovule was same. So what was the genetic constitution? If you just divide capital A, capital A, small a into capital A, capital B, small b, into capital A, capital B, small b. So we will have 4 different types. That is capital A, capital B, capital A, small b, small, capital B, and small a, small b. Similar will be for the ovules also. Now in this checkerboard, if we start filling or we put on this characters, we'll get to see that many of the seeds are coming round and yellow. Only 1, 2, 3 are coming yellow and wrinkled. Wrinkle means that is small b it is coming. And 3 seeds are coming which are round but green in color. That is capital A is not there. So it is small a, capital B. And only 1 seed we get which is green and also wrinkled in color. So that is small a, small b, means all are in recessive form. So if we take or if we see the seeds or what is the particular type we have got, we can see that round and yellow seeds are 1, 2, 3, 4, 5, 6, 7, 8, 9 of them are there. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So we can tell it is 9 of round and yellow, 1, 2, 3 wrinkled yellow or yellow and wrinkled, 3 of green or round and green, and 1 is green and wrinkled. So we get the phenotypic ratio, that is what we are able to see from here. It is 9:3:3:1. So this is the phenotypic ratio. But when we go for the genotypic ratio, the genotypic ratio like monohybrid cross was 1:2:1. Here the genotypic ratio is not so simple because we have different kinds of genotypic constitution. So here we will have 1, 2, 3, 4, 5, 6, 7, 8, 9 type of genotypic constitutions will be there in this checkerboard having a dihybrid, that is 2 characters in constitution, 2 characters, particular 2 traits have been studied. So we'll have 9 different types of genotypic constitution having a ratio of 1:2:2:1:1:4:1:2:1. So if you study this one and go about with this, that is capital A, small capital B, we have only 1 plant. Like capital A, small b, we have only 1 plant. Capital A, capital B, small b, we have 2 plants. This is the 1 plant, this is the 1 plant, 2 plant. So like that we can constitute or we can find out the genetic constitution of each plant. There will be 9 different types. So this gives the genotypic ratio of a dihybrid cross, and the phenotypic ratio is 9:3:3:1. Now, based on these experiments that what Mendel did, he formulated some laws. He formulated 3 laws of inheritance. So these laws are called the laws of inheritance. He formulated 3 laws, that is law of dominance, law of segregation, and law of independent assortment. So these 3 were taken, as the laws of inheritance which were formulated by Mendel based on his experiments. So first is the law of dominance. That is when 2 individuals are there, 2 individuals are crossed, the dominant trait is always expressed. The dominant trait is always expressed as the phenotype of the offspring, while the recessive trait remains masked. So when the 2 individuals of contrasting characters, that is tall and dwarf, that is round and green, that is smooth and wrinkled, if it is crossed, these 2 particular individuals of contrasting characters are crossed, then the offspring will show or will give always express as the dominant phenotype. The dominant phenotype will always be expressed and the recessive phenotype will be masked. So this was the law of dominance which we are told. Law of segregation. During the formation of gamete, each of the 2 allelic pair for a particular trait separates to form the genetic constitution of each gamete. When there is a formation of the gametes, always the 2 allelic pair separates out. Here you can see when we were talking of, The allelic pair was capital T. During the formation of gametes, so capital T became 1. Capital T, small t, during the formation of gametes, the alleles got separated out. One gamete was capital T and the other gamete was small t. The one gamete was capital T, so other gamete was small t. So during the process of formation of gametes, the allelic pair, the genetic constitution of the allelic pair, it separates out, and each of this allele goes into or gets into the gamete. And genetic constitution of each gamete, it forms the genetic constitution of each gamete. Law of independent assortment, this was the third law. During the formation of gametes, the segregation of one allelic pair is independent of the segregation of the other allelic pair controlling the trait. So when we were talking of the dihybrid cross, we told you that we had taken into consideration two traits, that is round and yellow seeds, green and wrinkled seeds. But when this round and green combined, the yellow and the green were influenced by the gene capital A, small a. And wrinkled and round was influenced by the gene capital B, small b. But this A and B gene did not interfere into each other. These two genes, that is capital A and capital B, capital A and capital B did not interfere into each other. Means this round gene, the gene for giving the round shape, did not interfere into the color of the seed, or the color of the gene for the color of the seed did not interfere into the shape of the seed. So we can tell that when the gamete formation takes place, that is during the formation of gametes, the segregation, that is segregation of one allelic pair, is independent of the segregation of the other allelic pair controlling the trait. So they are completely different. They do not interfere between each other. They are completely different, and during the gamete formation also, they separate out without any confusion. So these were the three laws that was formulated by Mendel based on his experiments of the monohybrid and the dihybrid cross. When we tell that sex determination, what does it imply? That when a boy or a girl is formed, we have already studied that the male gives the sperms and the female gives the eggs. But we knew that human beings are composed of 23 pairs of chromosomes, consisting of 22 autosomes and one pair of X chromosomes. Now the sex chromosome that is present for a human being or for a female, it is the X chromosome, whereas for a male it is X and Y chromosome. So when we told that during the segregation, the law of segregation, when we are talking of when the gametes are formed in a male, that is some sperms constitute or they carry the X chromosome and some sperms carry the Y chromosome. That is totally independent of each other. Who will be carrying X and who will be carrying Y, we are not able to determine. So some sperms carry X chromosome and some sperms carry Y chromosome. But the egg that is formed from the female always carry the X chromosome, because the sex chromosome for females always have X chromosome. They don't have Y chromosome, so they will be always, the egg will be having a uniform X chromosome always. So now when the sperm comes and fuses with that of the egg, now this sperm that fuses, it can be a sperm carrying an X chromosome or a sperm carrying a Y chromosome. So when we tell that what determines or what is the factor for sex determination or who determines the sex, that is, we can tell it is the male counterpart during a childbirth. It is the male counterpart which determines the sex chromosome, because the sperm is the only gamete which carries either X or Y chromosome. So if the Y chromosome goes and fuses with that of the egg, it is the X and Y, it produces a boy child. If X chromosomes comes and fuses with of the sperm, it is a X chromosome, so the ovule or the egg that is formed at its XX means it will give rise to a girl child. So it is totally dependent on the male sperm or the sperm which comes and fuses with the egg. Depend on what it will actually give rise. It is either a boy or a girl. So the sex determination is done by that of the boy, or it is done for male, because they are the carriers of the different types or the two types of sex chromosomes, X and Y. Female carries only XX chromosomes, so they cannot determine the sex. It is the males in the human beings who determines the sex chromosome, the sexes. So this brings about the end to the chapter, that is heredity and sex determination. In my next video, I'll be coming up with the questions. It will be mostly MCQ based because of the change in the exams pattern for CBSE. We'll be talking of MCQ based and very short answer type. It will be some cases MCQ also will be discussing. So that will be my next video. Till then, goodbye, stay safe. And if you have liked my video, please like, share and subscribe to my channel of sure. And if you have any doubts, any queries, please put in the comments box. I'll be surely getting back to you. Thank you.